Glycolysis takes place in the cytoplasm of every living cell. The glucose has to get into the cytoplasm across the cell membrane initially, and this can be via facilitated diffusion or via active transport. Both processes go through a glucose carrier protein to allow glucose in the cytoplasm. The glucose is then phosphorylated twice. This is the addition of two phosphate groups, which uses up 2 molecules of ATP, and it's called the investment phase. As the glucose becomes phosphorylated twice, hexose bisphosphate is formed, which is quite reactive and quite unstable. Hexose bisphosphate therefore splits into 2 molecules of triose phosphate. Each triose phosphate has a single phosphate group as part of the molecule. Two processes now happen to each triose phosphate. So triose phosphate donates a phosphate group onto ADP, and inorganic phosphate from the cytoplasm attaches to another ADP. So per triose phosphate, 2 molecules of ATP are produced. This is called substrate level phosphorylation. Substrate level phosphorylation does not require any electrochemical gradients. It just requires the breakdown of a substrate to release some energy to transfer a phosphate group directly onto ADP to make ATP. The second process to happen to triose phosphate is its oxidation. Oxidation is the loss of hydrogen, which is catalyzed by an enzyme called dehydrogenase. In the active site of dehydrogenase, triose phosphate has a hydrogen removed, which is then added to NAD, which is called a coenzyme, and we also produce NADH, the reduced version of NAD. The loss of phosphate and the oxidation of triose phosphate results in the production of pyruvate, which can go into the mitochondria if there is oxygen present, or if not, can be reduced to lactate in a muscle cell or a liver cell, or it can also be converted to ethanol and carbon dioxide in yeast cells or plant cells. The net products of glycolysis include 2 molecules of NADH, 2 net molecules of ATP, 4 are made, but 2 are invested, and 2 molecules of pyruvate.